Hello and a very warm welcome to everyone joining us for today's webinar titled Accelerate DNA to Therapeutic Product Timelines, Innovate Your Cell Line Development Process. My name's Jess Smith and I'll be moderating today's session. I'm delighted to introduce today's webinar speakers, Dr. Devika Kalsi and Dr. Xin Liu. Dr. Devika Kalsi is a principal scientist in the upstream process development team at Fujifilm Diosynth Biotechnologies, Billingham, UK, specializing in mammalian cell line development. Dr. Xin Liu is head of cell and molecular biology at Sphere Fluidics and is responsible for managing the development of assays and applications for Sphere Fluidics' flagship project, Cyto-Mine. In this webinar, they will share how scientists at Fujifilm Diosynth Biotechnologies have combined Sphere Fluidic Cyto-Mine technology with a plate imaging method to create a novel workflow for the generation of high-quality clonal cell lines. They will also illustrate how the use of this key technology has reduced timelines from 25 to 10 weeks. Following the presentations, we'll have a question and answer session. If you have any questions for our speakers, please feel free to submit these at any time during the webinar. You can submit your questions at the left of your screen. Simply hover over the purple icon and click to type in your question. Without further delay, I would like to hand over to our speakers for today's presentation. Please go ahead.
Okay, thank you, everyone. My name is Xin Liu. I'm the head of Cell and Molecular Biology Group at Sphere Fluidics. It is my great pleasure to be part of this joint webinar together with Devika, colleague from Fujifilm Diosynth Biotechnology. Today's presentation, the title of today's presentation is Accelerated DNA to Product Timelines through the integration of Cyto-Mine technology into Apollo X cell line development process. Now, majority part of this presentation will be covered by Devika, although I will kick off the session by providing you a brief overview of the Cyto-Mine technology. And then Devika will take over to showcase how they adopt Cyto-Mine technology to establish a robust cell line development process within a 10 weeks timeline. Okay, Cyto-Mine is an instrument platform used for single-cell analysis developed by Sphere Fluidics. It is a benchtop unit encapsulated a complete workflow for single-cell analysis. The key technology behind Cyto-Mine are all materialized in the so-called Cyto cartridge. So this is one piece of microfluidic subject embedded with all the microfluidic networks, functional units that required for complete workflow for single-cell analysis, starting from sample loading up to dispensing the single cells into the wells of microtiter well plate. I will explain the workflow in the next slide, but before that, I would like to highlight a number of attractive features of the Cyto-Mine technology platform. It used picodroplet microfluidic technology, which essentially enabled us to provide the user a seamlessly integrated workflow that make it possible, again, for us to provide extremely user-friendly operation environment. The software in Cyto-Mine has been designed to be extremely intuitive. The user does not need to carry out sophisticated fluidic or other parameter optimizations. They can just focus on their assay and biology. In addition to that, more importantly, there are a number of attractive features which can make Cyto-Mine a highly liked device for single-cell analysis for biotherapeutic discovery and development. It is a high-throughput technology allows you to implement various unique assays that cannot be easily implemented in conventional technology platform. It is a high-throughput technology allows you to process tens of thousands up to millions of single cells in a single screen. As you will see later, is supplied with a fluorescent detection capability to allows you to rapidly screen the single cells based on their fluorescent signal inside the picodroplet. It also been implemented with a monoclonality assurance functionality, which has been specifically designed for cell line development requirement. There's a built-in imaging system allows the system to capture high-resolution images of all the single cells being dispensed into the microtubule well plate. The user can then take those images as evidence of monoclonality of the single cells they have cloned into the microtubule well plates. Overall, its instrument platform has been carefully designed to meet the requirement of biotherapeutic research labs works. For example, the materials and reagent within the Cyto-Mine system has been carefully chosen to be animal origin free. The Cyto cartridge is a disposable consumable you use in your experiment. One project, one cartridge. Minimize the cross-contamination across your projects. Now, let me explain slightly in detail the workflow in Cyto-Mine. So it all starts from the user load about 1 milliliter of cell suspension together with assay reagent. You load your cell sample into the sample chamber on the cartridge, close off the lid, mount the cartridge into the instrument. All the subsequent operation steps will be carried out inside the instrument by the control software. So the first operation step involves generating millions of miniaturized water in oil droplet. So this is a step when you will flow the cell suspension in a highly controlled manner into a microfluidic unit inside the cartridge to achieve highly monodispersed, high-throughput droplet generation and cell encapsulation. As shown here, the picodroplet formed in the process is stable against coalescence. Even if they are densely packed in a closed array, they will not coalescence against each other. Simply due to the fact in the oil phase, we have dissolved a special chemical called Cyto-Surf, which will assemble onto the water and oil interface and protect droplet from coalescence. The next step involves incubate droplet for extended period of time to allow the molecule secreted by the cells to accumulate inside the picodroplet. So as shown here, all the millions of picodroplet generated during the encapsulation stage will be captured inside an incubation chamber inside the same Cyto cartridge. A heating element on the instrument will heat the incubation chamber to 37 degrees. So this is the stage when a single cell will start to secrete the antibody molecule, for example. And hold in mind, the size of a picodroplet is only several hundred picoliter in volume. So it is an extremely small volume compared to the liquid volume you have in a conventional microtubule plate. The consequence of that is that only after 1 to 2 hours incubation, the concentration of the molecules secreted by the cell will reach to detectable level already. You can then, using a fluorescent assay, to detect the antibody molecule secreted by those single cells. Next step, after the incubation, you can use a fluorescent assay to detect the antibody molecule secreted by a single cell by flowing the single picodroplet one by one through a laser detecting module. This is a stage when the system will interrogate the fluorescent intensity of each picodroplet. As an end user, what you'll get is a two-dimensional dotted plot as shown here. You can gauge on the population of interest to allow the system to select those picodroplets meet the fluorescent criteria you have set within the gate. After sorting is finished, the system will then move into the next stage called dispensing. So this is a step the system will dispense each of the collected positive hits into each well of the microtubule well plate. As shown here, there's a built-in camera inside the instrument which will capture 5 snapshot images of a pick droplet right before it is dispensed into a well of a microtubule well plate. The user can either prefill the plate with culture media or lysis buffer, depends on their application requirement, and mount into the instrument to receive each pick droplet being dispensed. So during the dispensing, when the droplet being ejected into the well containing the media, the cells inside the pick droplet can be released spontaneously back into the cell culture medium. Therefore, the user can further bring the plates to CO2 incubator to allow the cells to grow into colonies. So, an attractive feature of Cyto-Mine by carrying out the single-cell assay in miniaturized pick droplets, which is of interest for cell line development, is that it allows you to select the single cells based on antibody secretion. As explained, we can use a fluorescent assay to detect the antibody molecules secreted by a single cell. For example, a single CHO cell inside this picoliter volume droplet. So you can co-encapsulate this single CHO cell together with a pair of affinity probes that can potentially both bind onto your target molecule of interest. So one affinity probe will be conjugated with FRET donor fluorophore. The other one can be conjugated with FRET acceptor fluorophore. Now imagine if your cells are secreting the IgG molecule of interest, you'll get this 3-molecule complex, which will essentially bring the FRET acceptor and donor into close proximity. What you can then expect when the droplet passing through the laser spot, you will detect decreased donor fluorescent intensity and increased acceptor fluorescent intensity. Based on that, you can easily detect the antibody secreted from a single cell after only several hours incubation. Another attractive feature which is showcased in this model experiment is that using such a homogeneous FRET, you can potentially select the cells based on their productivity. So this is a model experiment carried out by, first of all, generate 6 population of pick droplets, with each of them containing different concentration of the target IgG molecule, whereas they all contain the same FRET signal. And the data is presented in this 2-dimensional scatter plot with x-axis shows the donor fluorescent intensity, and the y-axis shows acceptor fluorescent intensity. As you can expect, if a pick droplet containing high amount of a target molecule, it will appear at top left corner of the scatter plot corresponding to an increase in FRET acceptor intensity and decrease in donor fluorescent intensity, whereas the negatives will naturally appear at the bottom right on the scatter plot. So based on their location on the scatter plot, you will be able to judge the abundance of the target molecule inside those pick droplet population. Now, in the case when you carry out the screening using a real cell sample, as shown in the scatter plot here on the right, you encapsulate single CHO cells into a pick droplet population, and using FRET assay to detect their fluorescent intensity, you obtain a scatter plot shown here. A population densely packed population on the bottom right shows the pick droplet containing no cells or containing cells that are not producing antibodies. Whereas the population spread diagonally covers a wide range of threat response, those picodroplets containing single cells with different productivity. Hold in mind, a picodroplet population has all been incubated for the same length of period. So different threat response tells you the single cells in those picodroplets have different productivity. So based on this principle, if your gate on a population give you a high threat response, potentially you are enriching the single cell population with high productivity. So this could be a highly attractive feature for cell line development applications, where people not only want to clone the single cells, but also want to select the single cells with the best productivities. Another attractive feature in context of cell line development is that the system have a built-in imaging functionality to capture the image data of each picodroplet being dispensed into each well of microtiter well plate. Being able to image the single cells in a miniaturized picodroplet allows us to provide you with a high-resolution image of the cells inside the droplet. And also, as shown in the images, in the set of 5 images in the middle, this is a set of a snapshot from a single picodroplet while it flows downstream a microfluidic channel. So due to the internal flow or inside the droplet, which will constantly change orientation of the cells, that provide you extra opportunity to being unambiguously judge how many cells are there in this picodroplet. So the end point of workflow, as shown on the right, is a color map of the plates showing the number of cells in each wells. So the green wells will be flagged by the software as shown containing a single cells in there, whereas the red ones are those wells that's known has received a picodroplet containing more than 1 cell, whereas the yellow ones are those empty ones. So what happens was that after the experiment, we have a data analysis software package which will automatically scan all the images collected from the experiment across all the single picodroplet being dispensed, and make a judgment of the cell number in those droplets, and then color each well of the pile of plates you collected. The user can retrieve the data after the experiment. They can essentially click through each well to review the cell number images. So those image data can be selected and saved by the user to be supplied as evidence of monoclonality. Okay, before I pass on to Devika, just one thing I want to highlight is that picodroplet microfluidic technology is a generic platform that allows you potentially to implement various other applications other than cell line development. If you are interested to learn about more other applications of Cyto-Mine, you can access a repertoire of application notes and other information on our website. Now I'm passing on happily to Devika to carry out the rest of this presentation session. Thank you very much.
Hi everyone, my name is Devika and I work as a principal scientist in the CHO cell line development team at Fujifilm Diosynth Biotechnologies in the UK. We have a long-standing relationship with Sphere Fluidics and have been working with them on a number of projects in the past few years. We employ their Cyto-Mine technology in our Apollo X CLD platform. Following on from Xin's presentation, I thought it would be a good idea for me to sort of provide a case study on the application of the Cyto-Mine in CLD. So quick presentation overview. Today I'll be going through a quick overview of the Apollo X expression platform. I'll then go on to provide sort of the key bottlenecks that we face in cell line development and the considerations that we would need to take into account for optimizing single-cell cloning. Next, I'll go through how the adoption of the Cyto-Mine technology has helped us to establish a robust CLD platform process with a condensed 10-week timeline. So just a quick summary of Fujifilm as a company. Fujifilm are a world-leading contract development and manufacturing organization with over 25 years of experience in biologics, contract development and manufacture. We rely on three main technologies for the production of protein therapeutics: microbial, mammalian, and viral vectors. We currently also hold 18 licenses for commercial manufacturing. We're in a phase of rapid expansion at the moment, acquiring many sites in the US, UK, and also expanding into Denmark. So the first iteration that we developed for CLD purposes in Fujifilm was the Apollo platform. As a forward-looking company, as soon as we launched the Apollo system, we started work on the Apollo X platform. The main aim for Apollo X was to increase the titers but reduce the CLD timelines. There are four key components in the Apollo X system. Firstly, we've got the expression vector, which is random integration based and has a DHFR-based selection system. We normally get really high productivities from selection itself, so there's no need for multiple rounds of gene amplification. Next, we have a host cell line, which is one of the things we really wanted to improve upon, going from Apollo to Apollo X. We have a CHO DG44 derived cell line, which we have directly evolved to have superior growth and productivity characteristics. The cell line can grow well in chemically defined suspension culture. On the process side of things, the biggest change we made was the adoption of the Cyto-Mine, which meant we had a robust single-cell cloning method, which only needed one round of cloning and minimum screening afterwards to obtain a high-producing cell line. On the media side of things, we developed a new basal medium that was chemically defined and protein-free and can support higher titers. We can use this medium from CLD all the way up to GMP manufacture. So I'll just quickly go through the improvements that we made to our host cell line. As mentioned earlier, we used a technique called directed evolution to kind of evolve whole pathways to improve our host cell line. For Apollo, we looked at adapting our cell line to chemically defined suspension culture and to the stresses of single-cell cloning. For Apollo X, we kind of did the same. We had 2 rounds of directed evolution, but evolving it to different stresses. So we first evolved it to the stresses of a chemostat culture, and then moving on, evolved it against a subculture regime. What this resulted in was getting a cell line, a host cell line, that had a faster doubling time. It grew quicker, which meant we had a reduction in our CLD timeline already. As well, we sort of performed a few mini CLD projects with cell lines derived from the Apollo host and the Apollo X host, and we were able to show that the Apollo X host had, on average, a doubling of titer. The last thing, obviously, is it's good to have all of these advantages, but you don't want to have any negative impact. So we checked the product quality of the maps that were produced, and there was no negative impact, so that was a win. The main focus of this talk is to go through the CLD process. When we were looking to develop the Apollo X platform, we knew one thing we needed to balance was speed and the quality. As a contract manufacturer, the main thing you want is for your customers to get to quick and efficient IND submission and approval. You want them to get to that clinic as soon as possible, but you have to make sure you're still maintaining the high quality of cell lines that you get. A robust CLD process has cell lines at the end of the process that exhibit high titer, favorable product quality, and are stable, but that needs to be achieved through low screening efforts. You can't screen multiple rounds and then expect to have a cell line in a quick amount of time. If we delve further and break it down, we look at the single cell cloning step that forms part of CLD. We need to have a fast development process and balance that with regulatory compliance. So I've just mentioned single cell cloning. Let's delve further into that. Why is it important and why do we want to improve it? There is a regulatory requirement that recombinant cell lines need to be derived from a single cell. Traditionally, scientists would go about doing this through low cell density plating conditions known as limiting dilution cloning. What happens in limiting dilution cloning is that the method's really low throughput, so there is a requirement of lots and lots of plates, making it a labor-intensive process. As well, one round of cloning is not enough to give you a high probability of monoclonality, so you aren't that confident that you've got a monoclonal cell line. So essentially, you need to have at least 2 rounds of cloning, further elongating those CLD timelines. In the past decade or so, there has been sort of a shift in industry, moving away from the cumbersome LDC methods, looking into having a more image-based assurance of monoclonality, and there have been lots of technological improvements that have aided this shift in focus. Still, with having that monoclonal cell in a well image, you need to have a probability of monoclonality to show your process robustness. So this was sort of our starting point. Before we launched, or before we started work on Apollo X, this is what our Apollo platform looked like. We had 2 rounds of cloning and multiple screening steps contributing to an 18-week timeline. Taking into consideration the points I've just made, that shift in focus, we knew it was time to make a change to our cloning process. So we went back to the drawing board and started writing down what were the requirements, what were the factors that we needed to consider to make a new CLD cloning process. We needed that process, firstly, to be high throughput, especially comparing it to LDC, which is super low throughput. As well, a benefit would be if the process had single-use consumables so that we could go from project to project real quick and have that segregation between programs. On the regulatory aspect, which is probably the most important one, we needed the equipment or the technology to be regulatory friendly and have that imaging capacity. In the number of cloning rounds, obviously we wanted to reduce that one. We wanted to be able to have a single round of cloning instead of doing 2 rounds to save time. As well, having a screening capability embedded in that single cell cloning step, so having an indication of productivity right at the start would help reduce the number of screening steps you would have later on. And lastly, perhaps another important aspect is you need to have an equipment that would be gentle on your cells, not impacting your cell viability negatively, and that your cells should be able to recover from that cloning step. So after a lot of market research, a lot of trialing of different technologies, we decided the one that met our criteria was the Cyto-Mine developed by Xin Liu. We decided to couple the Cyto-Mine with a high-resolution plate imager, which meant we had double the monoclonality assurance, and because of the advantages of the Cyto-Mine, could have a high probability of monoclonality in 1 round of cloning. So this slide just demonstrates sort of a little workflow on how the user would use our single cell cloning step, how they would go about that in the lab. So you've got the Cyto-Mine, which is essentially step 1. And as Xin would mention, we would look at inputting a certain cell concentration into the machine. The single cells would then be encapsulated into single picodroplets, which can then be assessed individually. We can stain them to assess them for their productivity, and then kind of select the highest or the best producing cell lines to then dispense into a well in a 96-well plate. So 1 picodroplet gets imaged and dispensed into 1 well. As each picodroplet goes through the dispenser channel, it's imaged 5 times. So you've got a really robust and clear-cut image of your cell contained in a picodroplet. So that was step 1. Then we combined that with step 2, which we would image. So once the cell has been deposited into the well in a 96 well plate, we would then image the cell inside that well. So you've got an extra monoclonality image evidence. So we would do that on the Solentum cell metric, where we can image plates at multiple time points. So essentially, you've got your image evidence on day 0, on the day of seeding, with the picodroplet image and your cell metric image of the cell in the well, and then you can start tracking how that cell would develop into a colony when it would divide on day 1 into 2 cells and forming a full-formed colony on day 14. So in this way, we can see that the Cyto-Mine and the cell metric provide a multi-step image assurance of monoclonality. So it's all well and good, we've got a new system in the lab, but there's always going to be steps of optimization and fine-tuning for it to get the best results that we possibly can. So we started working with Phil, who firstly optimized the protocol. The first thing we needed to check in that was that our input cell concentration was the most adequate to achieve a good probability of monoclonality. What I mean here is the amount of cells you're going to put in the machine is going to impact how many cells get encapsulated and what percentage of that cells encapsulated contain, how many picodroplets would contain that one cell. The main thing you want to reduce is having polyclonal droplets, because it's easy to distinguish between an empty droplet and a droplet containing one cell, but polyclonal droplets would then lower your probability of monoclonality. Next, we looked at our cloning media, which is sort of a really important factor in getting that cell to recover and form a colony. So we looked at supplementing our basal medium. We looked at 5 different supplements that we'd found from literature and tried to combine them in a 2-level DOE factorial design to test their efficacy in different combinations. So at the end of that, we were able to isolate the combination that worked best for our cells and was able to support high levels of clonal outgrowth. At the end, we were able to have more than 50% cloning efficiency. The last thing we obviously wanted to sort of optimize was the use of that productivity screen. As Xin has mentioned, you can have different productivity assays or different sorts of assays that you can use to select for picodroplets in the Cyto-Mine. The one we used was the FRET Cytoselect assay, which can stain for productivity in map-producing cell lines. The advantage of the FRET is you're selecting cell lines based on their productivity, so you're essentially isolating through different gating strategies a set of high producers. We decided to compare that against a more random selection with a cell staining dye called Cell Tracker Green. Essentially, we ran 2 CLD programs in parallel with cells that were first isolated from the FRET, and then another set of cells isolated at random when you're just staining with the cell staining dye. At the end, when we ran these cell lines in an AMBER 15 FEDBAT screen, we could see that with the FRET selection, the cell lines that came through that method outcompeted the cell lines that came from the Cell Tracker Green method for title. One thing to note here is though you are seeing a little bit of a reduction in the Cell Tracker dye, on average, that reduction is there. However, you were still able to get a high title even from just staining with the Cell Tracker Green. So it gave us an option for molecules that were not compatible with the FRET assay to use a cell staining dye. So we've got a method now, we've got that image evidence, we've optimized it, we can get clonal outgrowth. So what's next? The last step in this puzzle is sort of trying to show your process robustness. What I mean by that is how confident you are in getting a cell line that is monoclonal at the end of cloning. For this, we use a calculation of probability of monoclonality. We went back to our process, we've got those 5 steps here, and looked at where exactly an error could occur, what could lead to a non-clonal cell line. We decided that the encapsulation step, the sort and dispense step, and the plate imaging step are the areas where you could have an error. We decided to calculate that error based on the Wilson score interval method, which we had verified with statisticians based in Newcastle University, and then we used that method to give us a single error event probability for each of the steps, and then combine it to give us an overall probability of non-clonality. When we looked back at our data for all the projects that we've run, when we combined all of that data, we found that the probability of monoclonality at the end of the process was always over 99%. So we're very confident in our method. These results and methodology has been verified as well in a peer-reviewed manner and has been published in the Biotechnology journal. So to summarize, the Cyto-Mine has been a massive key factor in helping us achieve a robust single-cell cloning method without compromising on quality of cell lines obtained. On the process side, we managed to get a method that was easy to use. The machine and the software are really easy to operate, so minimal training is required for any new starters. As well, we have the single-use option, so consumables in the Cyto-Mine are all single-use, so that turnaround between programs is maintained and that segregation is maintained. On the regulatory side, we were able to get that image evidence in the Cyto-Mine and the cell metric plate imager to have a multi-step image assurance of monoclonality. As well, the cell lines can be easily traced because you can use barcoded 96-fold plates as well in the Cyto-Mine. Perhaps the biggest advantage was on the practical end for us, because going from that 18 weeks, we were able to shave off 8 weeks of worth of time to have an 8-week reduction and a 10-week timeline for CLD. As well, having that integrated productivity screen, which can be used for mAb molecules, meant that you had a screening step already integrated in your single-cell cloning step, thus saving time as well. And lastly, the method is really gentle on the cells, and we've always got good viability and cell line recovery post-cloning. So the Cyto-Mine essentially ticked all the requirements that we had at the start. So to culminate all of our efforts, I just wanted to go through this last slide, which shows our track record for Apollo X. Through the use of the Cyto-Mine, improvements to the host cell line and the medium, we're able to have a 10-week CLD timeline. Apollo X was launched around 2 and a bit years ago, so by now we have now completed 24 CLD programs, 2 of which were non-mAb molecules. Generally, we have cell lines at the end in the fed batch amber screen obtaining more than 4.5 grams per liter in titer, with the highest commercial program achieving 6.5 grams per liter. So it just really goes to show how changes in different parts of the CLD process can have such an impact. So that's it from me. Thank you for listening. I just wanted to give a quick acknowledgement to the people helped on Apollo X. So all the people in the mammalian cell culture team at Fujifilm, especially Faye, Ali, Leon, Rachel, Nick, and Ellie. To the Newcastle University statisticians for helping us with our probability of monoclonality method. And obviously, last but not least, Therapeutic for always being there to help us with the development of the Cyto-Mine, the adoption of the Cyto-Mine, and always being on hand for any engineering support while we use it, especially Xin and Serena for always being on hand for technical advice and support. So thank you to them. And that's it from me. Thank you for listening. If you've got any questions, please let us know.
Thank you very much, Devika and Xin, for very interesting presentations. It's now time for our question and answer session. Please continue to send in your questions. You can do this by clicking on the purple Ask a Question icon to the left of your screen. Let's start with a question for Xin. The single cell secretion assay standard plot shows high overlap of signal for 2 versus 5 milligrams per litre of IgG. In cell line development, there are cases where 15% increase in productivity is highly economically significant. What is the minimum expression improvement that this system can reliably select?
Yeah, thank you. Good question. The in terms of the overlap of the scatter plot in that standard experiment, so there's a known phenomena when you do the binding assay called hook effect. So essentially, if your target molecule going above certain threshold, you'll start to see the drop of the assay signal. So that's why you see those two high concentration populations start to overlap. But what we see is that the incubation window and the assay dynamic range is good enough to allow us to reliably detect circulation from either CHO cells or in other cell types, for example, papilloma cells or primary B cells. The other question, I'm not sure whether Devika want to comment, but the only thing I can comment is that in terms of productivity or other features or value they want to gain from such a single cell technology, we see is highly diversified among the end user. So I think in terms of productivity enrichment is highly depend on their host cell lines, their track record of optimization of their platform, including the media, including the downstream, upstream various process. I think the bottom line is that Cyto-Mine is offer the user the opportunity to really to optimize the experiment conditions very easily. You don't need to have some specialized software package or procedures to, you know, uh, optimize your conditions. If you want, you can even customize your assay. So by doing this, I think the user can easily optimize the condition to give them the optimum results.
Lovely, thank you. This one is for Devika. Apollo-directed evolution. Were cells simply grown under the desired stressed conditions and relying on selection of spontaneous mutations or epigenetic changes to give the desired trait?
Thank you for your question. Essentially, yes. Yeah, we wanted basically to be able to manipulate sort of whole pathways in one shot. So yeah, we wanted to find out which cell lines or which of our host population would be most able to withstand the stresses and be able to outcompete sort of the unsuitable variants. So yeah, that was essentially the method we took to improve our host cell line.
Thanks. Another question, are there any other assays you can use for other types of antibody formats that you can use to select high producer cells?
Yes, in general, we have provide off-the-shelf assay kit called Cyto-Select that has been optimized to specifically detect human IgG or human fat fragments. And as I said, the advantage of using FRET is potentially the user can customize the assay for their specific molecule of interest. All you need to do is find a pair of recombinant probes that can both target on the protein of interest.
Okay, thank you. Please contrast Cyto-Mine with FACS for selection of high-producing cell lines.
Yeah, that's another very good question. Actually, as I just explained, our observation through various cell line development customers, the obviously people using various platforms, and the results are also very diversified. So our observation for the user using effects for selecting high-producing clones is a mixture. We do see people after years of optimization, they can achieve using flow sorter to achieve the high enrich the high titers. But I think our goal is to offer the user a fairly easy operation system, plus providing the extra information on the single cells being screened and cloned.
Thank you, Xin. Are you employing any advanced analytics during screening to interrogate direct product quality attributes such as glycan profile and other PTMs? Does this factor into your selection criteria in addition to titer?
I think that one's for me. So within Apollo X, we do evaluate the product quality at the Amber 15 stage. And we use analytics to look at the glycan profiles and things like high molecular and low molecular weight species. just check the purity of the product that we're getting and compare it against sort of the standard for that molecule. And then we do take that into account when we're selecting at the end of the cell line development process.
Thank you. A question here. What is the outgrowth of single cell cloning in 96 Wells plates?
So for us, for Apollo X, we get around 50% clonal outgrowth in a 96 well plate. So you're looking at around 14 days from that seeding of that cell in that plate to then have an outgrowth, have enough cells to then take forward into your next stage. So by that 14-day sort of standpoint, we have around 50% clonal outgrowth, and then we're able to take the cells out and kind of bulk them up to then go into that AMBER 15 screen.
Thank you. And another one here for Devika. How many Cyto-Mine runs do you perform in one CLD project?
So in one CLD project as standard, we would carry out 5 Cyto-Mine runs. So within that, each Cyto-Mine run would have one transfectant pool that is put into the machine to then generate cell lines derived from that transfectant pool. So you'd have a sort of diverse set of cell lines by the end of it, because they're coming from 5 different transfection events.
Thank you. And what is the max number of pico droplets you can evaluate in one Cyto-Mine run?
Right. In one Cyto-Mine run, the user have the option to choose the number of droplets they generated for the downstream process, ranging from about 1 million up to 2 million droplets.
In Apollo X, how many cell lines are evaluated at each stage, i.e. at Cyto-Mine and then at AMBER 15 and then in bioreactors?
That's a good question. So at the Cyto-Mine level, when we've got that outgrowth in the 96 well plate, we would evaluate up to 300 cell lines, and we'll evaluate them for titer. Then we'll move on. The next screen is the AMBER 15, where we'll take up to 48 cell lines into the microbioreactors to then assess for growth, product titer, and product quality over the 14-day fed batch process. And then by the end, when we're going to bioreactors, it's maximum of 3 lines that we would take forward, or if the customer has decided that they want this 1 cell line, we'll take the 1 cell line forward and do more experimentation on them.
Another question, if the fluorescence level is low after incubation, do you have to start the experiment again, or can you change the temperature back to 37 degrees C to incubate for longer?
Yes, you don't need to drop off the experiment. Basically, in the Cyto-Mine control software, it will allow the user to run a batch of droplets through the detector. If you found the assay signal is not sufficient, you can just directly going back to the incubation step to further incubate the cells to build up more signal. All you do is click a button on the control software and going back.
Brilliant, thanks. What technique do you use for the initial evaluation of titers before it goes to ambers?
So at the 96 well plate screen, we use the octet to assess for titer, so protein A generally.
Thank you. And a more general question, what do you see as the future of biopharma cell development?
I can comment from just the technology market perspective. I think there's a big task ahead of us is to be able to demonstrate a broader range of applications. So the bottom line is that being able to confine the single cells, 1 or 2 single cells in these miniaturized droplets, essentially opens the door for various new exciting application areas. As perfectly demonstrated by Devika here, you can confine the cells in there and build a high sensitivity assay to directly assay the cells on their secretion productivity. you can also potentially put two different cell types in this miniaturized environment to study the cell-cell interaction. For example, recently we demonstrated a number of new applications in Cyto-Mine involving either carry out a workflow for single-cell genome editing. We also demonstrated, you can co-encapsulate the CAR T cell with the tumor cells, essentially detect the either cell killing or activation of the T cells. So there's a very broad range of applications behind this. So that's what we are heading for, actually, just aiming for the demand in the therapeutic development requirement and of the user maximum flexibility in terms of assay implementation and user-friendly technology, so that, as I said, they can just focus on their biology and optimize their experiment parameter that they are most interested in.
Thanks. Did you have anything to add to that, Devika?
I think sort of in the biologics field, we are seeing kind of different sorts of molecules coming through now, so more complex ones such as the bispecifics. So I think kind of being able to monitor titer levels for those kind of molecules would be, again, something that industry is now focusing on.
Thank you. Other question, how much cell line specific optimization is conducted at the bioreactor development stage? What is the typical difference between AMBER versus bioreactor titer?
So for the AMBER, we use sort of a standard platform process, so standard media, standard feeds in-house that we've developed, and the standard fed batch process. And then the cell line will be subjected to the same process in the bioreactor, and we do get a good correlation between the AMBER titer and the fed batch bioreactor titer. At the end, it's up to the customer how much optimization they want, but that scale up is quite good in terms of going from the AMBER to a 10 litre or 200 litre fed batch in bioreactors.
Thank you. And one final question. Why has Apollo not made use of targeted integration, i.e. CRISPR-Cas9? Wouldn't this give greater speed and control?
That is a good question. In terms, I guess, with CRISPR-Cas9 targeted integration, you're targeting specific sort of sites or specific pathways. And with directed evolution, it may not be the most sophisticated technology out there, but it gives us kind of a bigger benefit when we've compared against targeted integration in terms of getting a larger improvement in growth in titer.
Well, thank you very much, and thank you for all those questions. That's all we have time for today. So thank you to Devika and Xin for today's informative webinar, and thank you to everyone joining online and for sending in your questions. I hope that you have found this a worthwhile session. If we didn't have time to answer your questions, we will follow up with these after today's event. If you have any other questions for Devika or Xin, you can email me at editor@selectscience.net, and I will pass your questions on. You can also download a certificate of attendance in the Related Resources tab to the left of your screen. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. That brings us to the end of today's webinar. Goodbye, and thank you once again for joining us.